Students, let's discuss the various types of problems you are asked regarding the inheritance of sex linked recessive traits. I may tell you that the sex linked recessive traits follow a zigzag pattern of inheritance, or we can also say that they follow a criss cross type of inheritance, or a diagonic pattern of inheritance, or a skip generation pattern of inheritance. So, what we mean by zigzag or by crisscross or diagonic or skip generation pattern of inheritance means that the males of first generation transfer their sex linked or X linked recessive traits to the third half of the third generation males through second generation females. That's why it is known as skip generation pattern of inheritance or diagonic pattern of inheritance. Now we should directly, I'm not going into detail of the what is skip generation pattern of inheritance or what is this or that. You have understood that already in the classroom. Now we should directly discuss about the questions you are asked in your NEET examination. For example, let us start by an example. For example, a, no, a hemophilic male marries. This is the sign used in genetics for male. A hemophilic male marries a normal female. We represent the hemophilic male by this is X hemophilic, Y normal. This is normal female, X normal, X normal. Now, during gametogenesis, we see what will happen. In one of the sperm, this X chromosome will go with H, that's hemophilic, this trait, this Y normal. Again, during meiosis, during the oogenesis, this X will get separated from this X. Now, further, we can evaluate it through the Print-Square ratio, or you can also call it as checkerboard method. This is over here, you will see males on this side, that's X hemophilic, Y normal. Again, this is X normal, X normal. Now, this X, if this X carrying sperm with hemophilic trait, it fertilized the ovum, it would be like this X hemophilic, sorry, X, this X hemophilic, X normal, X normal, Y normal, X normal, Y normal. So, in these females, you will see there is a hemophilic trait, but they are not hemophilic. They have a hemophilic trait, but they themselves do not have hemophilia, as they serve as carriers of this disease because they are heterozygous in heterozygous condition for the sex-linked recessive traits. So this is a carrier female, and again this is a carrier female, and these are normal males. So try to understand over here. Sometimes you are asked in your NEET examination a question like this, that for example, a hemophilic male marries a normal female. How many of the offsprings, or what percentage of the offsprings will suffer from hemophilia? You will see this is carrier female, this is carrier female, normal male, normal male. Among them, nobody is hemophilic, but these females do possess the hemophilic trait, but they themselves are not hemophilic. They can transfer the hemophilic trait to the next generation, but they themselves are not hemophilic because only a single allele for only a single allele has underwent mutation, but the next allele, the recessive allele in them, that's functional. Or we say that if one of the allele undergoes mutation, in this case The other allele is functional, so that these females are not hemophilic. For a female to be hemophilic, both the alleles must undergo mutation, and that is almost non-existent case. Now after that, let's attempt. This is parental generation. These are parents. This is parental generation, and this is F1 generation. Now let's assume that we will, for example, proceed forward with this example that let's a normal male, let a normal male, that is, this is the sign by which male is represented in genetics, that's XY, normal male. This is the sign representing male in genetics. If a normal male marries a carrier female, female is represented by this sign in genetics. Carrier female is that's X hemophilic, X normal. Now what will happen? During the gametogenesis, this X gets separated from Y, this X normal, this Y normal. This is X hemophilic, this is X normal. Again, now you can evaluate it through the Punnett ratio or by the checkerboard method. Over here, these are males on this side, male gametes, that's X carrying sperm, Y carrying sperm. This is X hemophilic O, and this is X carrying O. Always O has an X chromosome, but the sperm may either be androsperm if it carries Y chromosome, or it may be gynosperm if it carries X chromosome. Now you can evaluate it. X, this is X normal, X hemophilic, this is X normal, X normal, this is X hemophilic, Y normal, this is X normal, Y normal. This is carrier female. This female has a trait of hemophilia but is not hemophilic, has a trait of hemophilia but is not hemophilic. This is normal female. This has no hemophilic trait. This is normal male. But over here you will see this male over here. This male is hemophilic male. As you see, this male, in case of males, there's XY pair of sex chromosomes, and upon there's only single X chromosome. Upon single X chromosome, there's a single allele for the formation of the coagulation factors, and if that undergoes mutation, there's no homologous allele upon the Y chromosome for regulating the synthesis of the coagulation factors, because Y chromosome is not a homologous chromosome of the X chromosome. So over here, for the synthesis or for the formation of the coagulation factors, there's only a single allele which we call as homozygous condition. When for a gene there is a single allele, we call it as homozygous condition. Okay. Now you will see, first of all, we shall go to this position. These are parents. This F1 generation. Now you should move on to this. This is F2 generation. Now again we will go to this parental generation. You see over here the male is hemophilic, the females are normal, so the trait of hemophilia is in the male. In the F1 generation you will see the females have the trait of hemophilia but they are not hemophilic due to their heterozygous condition. The males are normal. In other words, we say that in F1 generation nobody is hemophilic, but the trait of hemophilia is present in the carrier females. Now we should move on to the third generation. You see, in this generation you will see this is carrier female, this is normal female, means 50% of the females are carriers for the hemophilia and 50% are normal. This is normal male, means 50% of the males in the F2 generation are normal and 50% are hemophilic. So again we see that the hemophilia from first generation males gets transferred through the carriers of the second generation to half of the males in case of third generation. So we say that the trait of hemophilia gets transferred from first generation males to the half of third generation males through second generation carrier females, and that is why this type of inheritance is known as diagenic pattern of inheritance. By diagenic we mean dia means through, gynic means females, means the trait of hemophilia gets transferred from first generation males to third generation, half of the third generation males through second generation carrier females. It's also known as skip generation pattern of inheritance, as in F1 generation none of the individuals suffers from the hemophilia, although the females are carriers for the hemophilia. It's also known as skip generation pattern of inheritance, or it can also be called as zigzag pattern of inheritance or crisscross pattern of inheritance. Hope that you have understood the crisscross pattern of inheritance. Now, I may ask you something more about this. Sometimes you are asked about the ratios. For example, take the example over here. A hemophilic male marries a normal female. The question may be, if a hemophilic male marries a normal female, what would be the ratio of the carrier individuals to the normal individuals? It would be 50 to 50, or it would be 1 is to 1. Or you can also be asked like this: if a hemophilic male marries a normal female, how many of the offsprings in the F1 generation will suffer from hemophilia? None. Although these are carriers for the hemophilia, but they are not hemophilic. Similarly, you can be asked a question in this case: if a normal male marries a carrier female, what would be the ratio of normal males to the hemophilic males? There is one normal male, one hemophilic male, that is 1 is to 1 ratio. Or you can also be asked like this: if a normal male marries a hemophilic male, what would be the ratio between normal females and carrier females? There's one normal female, that's one carrier female. It would again be, the ratio would again be 1 is to 1. You can also be asked like this, that if a normal male marries a carrier female, what would be the ratio between the carrier female to normal female to hemophilic male to normal male? There is one carrier female, there is one normal female, there is one hemophilic male, there is one normal male. So the ratio would be 1 is to 1 is to 1 is to 1. Sometimes the question is asked in this way, that if a normal male marries a hemophilic male, how many carrier females would be there out of the total females? You have to answer it, that is 50% of the females would be carrier females. And you can also be asked that if a normal male marries a carrier female, how many of the males will suffer from hemophilia? This will again be 50%. So by this you might get a clue regarding as to how the questions are asked in a numerical sense regarding the skip generation pattern of inheritance, or regarding the diagenic pattern or zigzag or crisscross pattern of inheritance.